Welcome to another Royal Astronomical Society's online public talk. Hello, I am Lucinda Offer, Education Outreach and Bicentenary Events Officer for our 200th anniversary of the Royal Astronomical Society. Our organization advocates for astronomy, geophysics, and space science in the UK since 1820. I'm excited to introduce to you today our bicentenary talk guest speaker, Rosie Cane, who will speak with us about microbes and how we can use bacteria for the search for life beyond Earth. Rosie Cane is a final year PhD student at the University of Edinburgh and UK Centre for Astrobiology. She completed her undergraduate degree in observational astronomy at the University of South Wales, where she specialized her final year dissertation on the use of terrestrial extremophilic organisms as an analog for potential life elsewhere in the solar system. Her work focuses on bacterial responses to multiple extreme environments and understanding the biochemistry of life. She also has a passion for inclusivity in STEM subjects, outreach and public engagement and promoting physics and astronomy to GCSE level for females. This is all extremely interesting, especially with 3 launches scheduled next week for missions to Mars, 2 of which will be looking for life on another planet and also studying the atmosphere of Mars. Today's talk may give us a better understanding of what the scientists operating those rovers remotely will be looking for. And a quick mention for next week before we get started, the Royal Astronomical Society is working in collaboration with the University of Leicester's astronomy department called Chasing at Shadows, a live stream telescope observation from the summit of Mauna Kea. You can register to be with us. We'll be streaming live from 11 a.m. to 4 p.m. And we'll also have some guest speakers, Professor Emma Bunce, giving us an update on Jupiter, talking about the future JUICE mission and also Juno. We'll be live streaming from 11 to 4 all day, talking about the experiment and the science behind watching Ganymede transit the face of Jupiter. A quick reminder for today's speaker that there will be a Q&A after the talk, so please get your questions ready. There are places to post your questions for our speaker. If you've joined us on Zoom, there's a Q&A box, and I see someone's already used it. Well done. And on YouTube in the chat box. So if you're joining us on YouTube, you are definitely welcome to leave us some questions there for our speaker that I will refer to. So without anything else further, I want to thank our speaker, Rosie Kane, for being with us today and celebrating with us our 200 years of the Royal Astronomical Society. On to you, Rosie. There we go.
Right, so thanks, Lucinda, and a huge thanks for everyone at the Royal Astronomical Society for inviting me to give this talk, and a very happy bicentenary to them. Let's just make this. Yep. So good afternoon, everyone. Well, good evening now, everyone. Sorry, I did a talk this morning. My name is Rosie. I'm a final year PhD student at the University of Edinburgh, researching in astrobiology. I'm going to give you a brief and hopefully interesting introduction to how we can use bacteria in the search for life beyond Earth. Now, just for background, I'm an astronomer by undergraduate degree and have trained in microbiology over my PhD. So hopefully there'll be a good mix of both biology and astronomy in this talk. But with that, let's begin the virtual seminar. So in this talk, I will cover a introduction to astrobiology, a summary of what is life, what extreme environments are, and how they can help us assess Martian and icy moon habitability. And finally, how are we searching for life beyond our own solar system via exoplanets? And a good place to begin this talk is to define the term astrobiology. NASA defines astrobiology as the origin, evolution, distribution, and future of life in the universe. and it's an extremely multidisciplinary subject involving astronomers, biologists, chemists, geologists, physics, engineers, and many others, all working together to help us further our understanding of the conditions of other planetary bodies and their potential for life. Now, our planet orbits just one star of about 200 billion stars in our galaxy, in a universe of maybe 150 billion galaxies. With the universe so vast, astrobiology seeks to answer the age-old question: are we alone? Are we alone in the universe? And this question can be exciting at the possibility of discovering new life, but equally terrifying. In the search for life in a weird and wonderful universe, we need to know what to classify as alive. Many organisms beyond our planet, and indeed some found in extreme environments of our own planet, will seem, for lack of a better term, very alien to us. But from our understanding of life here on Earth, we're able to come up with some key factors that help us to define what life is. Now, defining life is not an easy task, but generally in astrobiology, we tend to go by the Darwinian definition of life, as NASA, which states that life is a self-sustaining chemical system capable of undergoing Darwinian evolution. And by Darwinian evolution, we basically mean that all species of organisms arise and develop through natural selection. They do this using small inherited variations that increase the individual's ability to compete, survive, and reproduce, as introduced by Charles Darwin. Going by this definition, it's immediately evident that by studying life, which is capable of surviving comparable environments here on Earth, it will allow us a certain level of predictive power when discussing the possibility of life on other celestial bodies. In addition to the definition of life, we also need to establish what life on Earth has in common. All life on Earth is made of what we call CHNOPS elements, so that's carbon, hydrogen, nitrogen, oxygen, phosphorus, and sulfur, with carbon being the backbone of most biological molecules. Life also requires energy to grow, able to adapt to its environment, can reproduce, can move, and can respire. And life on Earth is split broadly into three domains, which can be seen in the image on this slide. This shows the relation between the domains of life, rooting from the first primordial form or last universal common ancestor. The three domains we see are archaea, bacteria, and this huge section here, which is eukaryotes. And you can see that eukaryotes is actually split into lots of smaller sections. So it's actually split into plants, animals, fungi, and protists. And we also have humans up here in this corner. As we're looking at microorganisms in this talk, we can focus on archaea, bacteria, protists, and fungi. And if we look closer at some of these microorganisms, the most common that we actually find on the Earth are bacteria, viruses, algae, fungi, protozoa, and archaea. And some, like algae, can ruin a fish tank, some, like fungi, can be great on pizza, and others can send the entire world into quarantine. Now, all of these are microscopic, which means we need a microscope to be able to see them, with the biggest microorganism being between 0.1 and 0.3 millimeters, which is still pretty small. Microorganisms are unicellular, which means they're made up of one cell, with the exception of viruses, which are not actually made out of cells, but single particles called virions. Most commonly in astrobiology, we tend to focus on archaea and bacteria, in some of the harshest environments on Earth, known as extreme environments, environments too harsh for humans to survive in. So why do we use microbes in the search for life beyond Earth? Well, microorganisms are some of the most hardy and most importantly adaptive organisms found on Earth. Due to their simplicity and rapid rate of reproduction, forming millions of cells in a very short time, this allows for a large amount of mutation and adaptation, leading to microorganisms surviving and even thriving in some of the most extreme environments found on planet Earth. These environments are teeming with microbes that can survive the harsh environments. The organisms that inhabit extreme environments tend to fall into two categories: extremophiles, or extreme tolerant microorganisms. Extremophiles, from the Greek word philos, or love, not only grow under extreme environments, but thrive within them, and actually require specific extremes, such as pH, temperature, or salt concentrations to grow. Whereas extreme tolerant organisms are organisms not actually optimally adapted to these extreme environments, but that are still able to grow within them. And archaea is one of the main groups to thrive in these extreme environments. Although members of this group are generally less versatile than bacteria and eukaryotes, they're generally quite skilled at adapting to different extreme conditions, holding frequently extremophilic records. Some archaea are the most extremophilic organisms known. For example, the archaeal organism Mythanopyrus candeleri, strain 116, grows at temperatures of 122 degrees Celsius. That's the highest ever recorded temperature. Whilst the genus Picrophilus includes the most acidophilic organisms currently known, with the ability to grow at a pH of 0.06. Now, when we're talking about life in extreme environments, it's also worth bearing in mind that we tend to talk from an anthropocentric, so human-based point of view. As if microbes could express their views, they would call us extreme, as they would not be able to survive in the conditions we do, for example, your home office environment. Now, extremophiles were first discovered in these environments in the 1960s, within Octopus Springs in Yellowstone National Park, a series of alkaline hot springs. An organism called Thermus aquaticus was found here that could withstand the pH and temperature stresses of the environment. And since then, we've discovered microbes in lots of crazy environments on Earth. For example, sticking to Yellowstone National Park, this is the beautifully coloured Grand Prismatic Springs, named after its rainbow colours. And this is the poster boy of astrobiology and extreme environments, with each ring around the lake inhabited by different types of pigmented, temperature-loving microbes. And you can see just how large this environment is from the size of the people on the boardwalk. So this here is just one example of an extreme environment, but there are many more out there. For example, the depths of Earth's oceans or the interior of volcanoes. And many extreme environments actually expose more than one environmental stress at a time. These environments with more than one stress at a time are called multiple extreme environments and are really important in assessing the habitability of an environment. Let's take a look at some of these with relevance to Mars. So very often, studies in extreme environments tend to focus on just one singular stress at a time. For example, the low temperatures on Mars, which go as low as minus 150 degrees Celsius. However, we don't know how the addition of more than one of these stresses in an environment affects how bacteria grow. For example, the high amounts of UV radiation present on the Martian surface, the lack of a substantial atmosphere, and the toxic salts on Mars, including perchlorate salts, which are highly oxidizing and so explosive they're even used in rocket fuel. So to really be able to assess the habitability of an environment on Earth or beyond, we need to be able to understand life's ability to survive in multiple extreme environments. And multiple extreme environments are a cause close to my heart, as they've actually been the focus of my PhD. I've been looking at how combinations of extreme stresses affect how bacteria grow. And I won't go into too much detail about my PhD, as that's not the focus of this talk, but experiments were carried out in a lab looking at the habitability of aqueous environments on Mars and icy moons. These involve looking at combined stresses of pH, salinity, nutrient limitation, and temperature. By looking at how these extremes interact in an environment, we can improve assessments of the habitability of other planetary bodies. So from all of these stresses covered on Mars, it doesn't appear to be a very friendly habitat to life on the surface due to this multitude of stresses. But there is hope, as the subsurface of Mars appears to have more favourable conditions for life. This is as the subsurface is shielded from the harsh radiation of the surface, so may be more favourable. But why is it that we focus on Mars in the search for life beyond Earth? Well, Mars has been a major talking point since the 1800s. At that point, Italian astronomer Giovanni Schiaparelli, a director at the Brera Observatory in Milan, began mapping and naming areas of Mars. He observed areas that looked like channels, calling them canali. Now, this was misinterpreted over the years to mean canals, indicating some form of intelligent life on Mars to build such structures, like here on Earth. And this idea was further backed by astronomer Percival Lowell, who believed that these lines on Mars were actually canals used by the Martians to carry water down from the polar caps down to the equatorial regions. Now, sorry to disappoint, but as Schiaparelli was well aware, this wasn't the case. The Mariner 4 spacecraft imaged Mars in 1965, showing no evidence of this intelligent life on Mars. And since we've discovered that these so-called canals are actually riverbeds on Mars. So we still get an exciting finding from this, as that indicates that there was once stable liquid water on the surface of Mars. And there have been lots of exciting missions since the Mariner 4, and there are even more exciting ones to come in the near future too. We have the European Space Agency Roscosmos ExoMars mission, which has sadly now been pushed back to 2022 from this summer. But when this does launch, this will have the primary goal of being able to determine if there's ever been life on Mars and to better understand the history of water on the planet. The ExoMars rover, named Rosalind Franklin, includes a drill to access the subsurface of Mars to be able to tell us more about the habitability of that environment. We also have the NASA Mars 2020 mission, which is still currently scheduled for launch between the end of this month and August. The Perseverance rover will seek signs of ancient life and collect rock and soil samples for possible return to Earth. In addition, as Lucinda mentioned, we have another few missions that are supposedly launching this month as well. We have the HOPE mission from the United Arab Emirates to understand more about how Mars lost its atmosphere. And also the Tianwen-1 launching from China's National Space Center, including an orbiter, a lander, and a this mission is actually a lead to a proposed sample return mission in the 2030s. Now, all of these missions are very exciting, but getting to space is expensive. Is there a way we can study the habitability of Mars from here on Earth? And the answer is yes. We can use analog environments, which are environments that recreate some of the conditions we believe are present on Mars. And one of these is the Basque Lakes, a series of small pools filled with high concentrations of magnesium sulfate, Epsom salts, and known as brines. Because of the high concentrations of these salts we find on Mars, this represents an excellent analog site to the Martian surface. And you can see here we have individual brine pools, each surrounded by a magnesium sulfate salt crust. And I was lucky enough to visit this environment during my PhD for fieldwork. And of course, we took a plushy Mars to the Martian analog site. And now to tie this back in with the title of the talk, how can bacteria on Earth help us with the search for life on other planets or moons? Well, one way is by using these analog environments to find bacteria that can survive conditions we know of on Mars or in icy moons. We can then try and recreate some of these conditions in a lab to see how the conditions can affect how and if bacteria grow. We have a multitude of these analogues on Earth, seen in this figure, which match to different parts of Mars, so we can explore how its geology may affect habitability. These environments include Iceland, which is a highly volcanic region that can be an analogue to the volcanic Tharsis regions on Mars. The Danakil Depression, which is a geothermally heated environment and is actually one of the hottest environments on Earth during the summer, that we think could be an analogue to past conditions on Mars. We have the Basque Lakes, which I previously mentioned, Kangerlussuaq, which is an area of permafrost in Greenland, which we believe could be used as an analogue to the poles on Mars. The Makgadis salt pans, which were once a really salty lake, where all of the water evaporated and left only salt deposits behind. And the Rio Tinto River, which is blood red in colour and has some of the similar salts and minerals to those detected on the Martian surface. And all of these are very exciting environments, and can hopefully tell us more about the habitability of past and present Mars. So following this, what would potential life on Mars need to be tolerant to? Well, first, it would have to be able to survive the high levels of ultraviolet radiation due to the lack of a substantial atmosphere on Mars. It would also have to survive the low temperatures, have to survive an alkaline soil with pH indicated by the Curiosity rover in 2008 to be alkaline, and high concentrations of many salts, including these chlorate salts, which are found in rocket fuel. But on from Mars, as there are many places to look at in the search for life, and on further out into the solar system. There are several places in the outer solar system we think are promising in the search for life beyond Earth. These include the icy moons of the gas giants. There's the moon of Saturn, Enceladus, and the moon of Jupiter, Europa, which are both icy moons thought to have liquid water oceans beneath their surface. Few worlds in our solar system are as compelling as Saturn's icy moon, Enceladus. A handful of worlds are thought to have liquid water oceans beneath their frozen shell. Enceladus actually sprays its ocean out into space, where a spacecraft can sample it. From these samples, we've actually determined that Enceladus has most of the chemical ingredients needed for life, and likely has hydrothermal vents spewing out hot, mineral-rich water from its ocean. With its global ocean, unique chemistry, and internal heat, Enceladus is a promising lead in our search for life on other worlds. And in the search for life beyond Earth, we can't forget Jupiter's moon, Europa. So decades ago, science fiction offered a hypothetical scenario. What if alien life was thriving in an ocean beneath the icy surface of Jupiter's moon, Europa? The notion pulled Europa out of obscurity and into the limelight where it's remained, stoking the imagination of people both with and without the science community, who fantasize about humans discovering life beyond Earth. That fantasy, however, may be grounded in reality. Ground-based telescopes on Earth, along with the Galileo spacecraft and space telescopes, have increased our confidence for a surface liquid water ocean on Europa. We think that Europa's ice shell is 10 to 15 miles thick, floating on an ocean 40 to 100 miles deep. So while Europa is only 1/4 the diameter of Earth, its ocean may contain twice as much water of all of Earth's oceans combined. This does raise a bit of a problem, however, as if we observe life on Earth, it was long believed that the life cycle required sunlight in order to be sustained, a source of energy. With the depth of the oceans and the thickness of the ice covering Europa, there could be no chance of development of life, right? Well, in 1977, while exploring an oceanic spreading ridge near the Galapagos Islands, scientists discovered hydrothermal vents. To their amazement, they also found that the hydrothermal vents were surrounded by large numbers of organisms that had never been seen before. And these organisms were drawing their energy from the hydrogen sulfide gas pouring out of these vents. Following this, if a similar mechanism exists at the bottom of Europa's ocean, then life could be a real possibility, surviving using similar mechanisms. And we can summarize the main stresses encountered in icy moons would be low temperatures, high levels of salt, capable of keeping the ocean's liquid at such low temperatures, and high pressures caused by the thick ice layer above. As these moons are just so far away, there are analog environments on Earth we can use to study them. And one of these is Lake Vostok, a subglacial lake in Antarctica, which represents a good analog to the icy moons. The temperature of this lake is approximately -2 degrees Celsius, but the lake remains liquid due to a 2-mile thick ice sheet above it, exerting high amounts of pressure and keeping the water liquid. If any life survives in this environment, it would have to depend on a different energy source from sunlight. It's believed that organisms may get their energy from chemicals and rocks in the environment, or there may be hydrothermal systems present, just like those detected in the Galapagos Islands. Now, the best way to determine if there is life on Enceladus or Europa is just to go there. There are currently no planned missions to Enceladus, but there are several upcoming missions to visit Europa. These include the European Space Agency JUICE mission, the Jupiter Icy Moon Explorer, which is due to launch in 2022 and will fly by Europa alongside the other moons of Jupiter, Ganymede and Callisto. Then after this, the Europa Clipper is due to launch in 2024 to investigate and confirm the surface conditions on Europa. And this mission will hopefully be followed by the NASA Europa lander, which is still not a confirmed project, but a lot of work has gone into. The Europa lander would allow us access to Europa's ocean to be able to analyse the conditions and understand its potential for life. Now, there's another moon in our solar system that's big in astrobiology and has potential for life, and this is Saturn's moon, Titan. Titan is an extraordinary and exceptional world. Among our solar system's more than 150 known moons, Titan is the only one with a substantial atmosphere. And of all places in the solar system, Titan is the only place beside Earth known to have liquids in the form of rivers, lakes, and seas on its surface. Although this might sound promising for life at first, these rivers, lakes, and seas are actually made of liquid hydrocarbons like methane and ethane. And the Huygens probe landed on Titan in 2005, discovering low temperatures minus 180 degrees Celsius and confirming the presence of these liquid features. We do think that Titan may have a subsurface water ocean, like Europa and Enceladus. So perhaps Titan's subsurface water could be a place to harbour life as we know it, while its surface lakes and seas of hydrocarbons could conceivably harbour life that uses different chemistry to what we're used to. That is, life as we don't know it yet. Titan could also be a lifeless world, but that's not very exciting for an astrobiologist to think about. So with optimism, let's think about what kind of life might be able to survive on Titan. Now, there is a large amount of methane on Titan, which is sadly not due to these creatures, as it is on the Earth. But we think that maybe life could survive similar to the way that methanogens do on Earth. And methanogens are archaea that produce methane as part of anaerobic respiration. And these could plausibly survive on Titan. Although all living things on Earth, including methanogens, use liquid water as a solvent, it's conceivable that life on Titan might instead use a liquid hydrocarbon, such as methane or ethane. The fact that Titan appears to have lakes of liquid ethane and methane on its surface, as well as these rivers and seas, suggests that it could support hypothetical non-water-based life. And it has been speculated that life could exist in the liquid methane and ethane that forms these rivers and lakes on Titan's surface, just as organisms live on Earth in water. And such hypothetical creatures would take hydrogen in place of oxygen, react it with acetylene instead of glucose, and produce methane instead of carbon dioxide. By comparison, some methanogens on Earth obtain their energy by reacting hydrogen with carbon dioxide, producing methane and water. And to find out more about the habitability of Titan, the Dragonfly mission will sample and examine sites around Saturn's icy moon. The mission will launch in 2026 and arrive in 2034. The rotorcraft, designed to look like a dragonfly, will fly to dozens of promising locations on Titan, looking for prebiotic chemical processes common to both Titan and Earth. Dragonfly also marks the first time that NASA will fly a multi-rotor vehicle for science on another planet. And you can see it has 8 different rotors and it flies kind of like a large drone. And Titan can also act as an analogue to the very early Earth and provide us with clues as to how life may have even arisen on planets. During its mission, Dragonfly will explore diverse environments from organic dunes on the floor to an impact crater where liquid water and complex organic materials may have existed together for possibly tens of thousands of years. Its instruments will study how far prebiotic chemistry may have progressed and will investigate the moon's atmospheric and surface properties and its subsurface ocean and liquid reservoirs. But additionally, and most importantly, the instruments will also search for chemical evidence of past or extant life. And finally, to zoom out beyond our solar system and take a look at exoplanets, planets that orbit stars other than our own sun. The James Webb Space Telescope, now due to launch in March of 2021, will be able to tell us more about potentially habitable worlds outside of our solar system. Webb will tell us more about the atmosphere of extrasolar planets and perhaps even find the building blocks of life elsewhere in the universe. And it will do this using a technique called spectroscopy. Spectroscopy is simply the science of measuring the intensity of light at different wavelengths. The graphical representations of these measurements are called spectra, and they're a key to unlocking the composition of exoplanet atmospheres. When a planet passes in front of a star, the starlight passes through the planet's atmosphere. If, for example, the planet has sodium in its atmosphere, the spectrum of the star, added to that of the planet, will have what we call an absorption line in the place of the spectra where we would expect to see sodium. This is because different elements and molecules absorb light at characteristic energies. And this is how we know where in a spectrum we might be able to see the signature of sodium or other elements if they're present. And the James Webb Space Telescope will not only look out beyond our solar system, but will also analyse locations a bit closer to home, as it will investigate the plumes of Enceladus and Europa a bit further. However, the ultimate goal of the James Webb Space Telescope is to find a planet with a similar atmosphere to that of the Earth and detect elements indicative to life, such as those Schnapps elements. So with that, I hope that you've enjoyed a quick tour of how we can use microbes in the search for life beyond Earth. If you're interested in more astrobiology goodness, the Royal Astronomical Society Supermassive podcast has a new upload this week about looking for alien life, featuring Professor Jane Greaves from Cardiff University, which is really interesting, and you can find it on the Royal Astronomical Society website. So with that, I hope you've enjoyed this quick guide to astrobiology and how we can use bacteria in the search for life beyond Earth. And thank you for listening, and I'm happy to answer any questions.
Wonderful. Thank you so much, Rosie. That was great again to watch and listen again to your, you actually updated your talk this time at 6 o'clock. It was a good yeah, it's great to see that with, I know mentioning China, there's not a lot to know about it. But yes, if the audience, the attendees who are here with us on Zoom, if you want to go ahead and put your questions in the Q&A box, I'd be happy to forward them on to, and she'll answer them live here for you. Yeah, there's not a lot of information coming out from China other than that it's a 3-part mission, and it's really exciting. I've heard they're going to look for life, so that was part of the 2 missions. So NASA's Perseverance rover caching any samples that they're going to drill for. And I believe I've heard that China's mission is also going to be looking for life, but it's such a huge mission. I mean, putting an orbiter, a lander, and a rover. I mean, I think it would be just really great for China to successfully, land something on the surface of Mars. India was also planning on doing a mission to Mars. I think they're doing it, they're going to launch in 2024, so they obviously need more time. But India's first mission to Mars was an orbiter, and that was very successful on first launch, which is just fantastic for them. So I hope the same for the United Arab Emirates. The United Arab Emirates. This is their first launch to Mars, also an orbiter being launched by Japan. And I hope for them that first time it's a very successful mission. And you did talk about many other planets and celestial bodies in the solar system. I'm sorry to be so Mars oriented, but it's kind of hard not to, being a huge fan of Mars. And this talk on extremophiles, extremophilic organisms is a huge popular subject in science research right now. Obviously with these missions to Mars and other places around the solar system, but also as a field of study. So astrobiology is just growing as a field of study. So I'm really excited to see what's going to be going on with the UK. And especially since the UK does participate in creating some of the instrumentation for these rovers. They helped out with InSight. I'm not sure how much they might have helped out with well, obviously with ExoMars, they've helped out with ExoMars, with Rosalind Franklin for a big part. So, but I'm a little disappointed. We have to wait another 2 years, but that's what happens with these missions. They are, like Rosie said, they take a long time. They're very expensive, and you really want it to be a successful launch. So unfortunately, ExoMars has been delayed for a couple of years. Okay, so we have a few questions. Actually, they're starting to grow now. So good. I've talked enough so that people can catch up with their questions. I'm just going to start us off. Thank you, Yvette, for joining us as an attendee, and she just wants to thank you, Rosie, for your talk. Amy Solomon asks, here we go, what made you want to pursue a career in astrobiology, or what is, specifically maybe your line of research? And she says, thank you.
So I studied astronomy at undergraduate level. So in my first year, astrobiology was actually an option for a course that I could take. And I really enjoyed it. So I looked up whether you can pursue things in astrobiology beyond an undergraduate degree. And it turns out that you can. So yeah, I applied for a PhD at the University of Edinburgh with Charles Cacao, and that's currently where I'm working, finishing up my PhD thesis. And yeah, very much enjoying it. And my research, I think I pretty much covered it in this talk. It's basically looking at how bacteria respond to these multiple extreme environments to help us understand the habitability of other planets and moons.
I wanted to ask you, working at the UK's Astrobiology Centre, what kind of environment does that afford you as a researcher?
There's definitely a lot of exciting opportunities. So we're based in Edinburgh, but we have a underground lab facility in Whitby in Yorkshire. So experiments can be undertaken underground. There's lots of opportunities as well for collaborations with lots of people. It's affiliated with NASA, so there's lots of chances to get involved and lots of conferences to attend and things. So it's been a great opportunity.
Yeah, a good way, definitely, to network, and I'm sure the group of astrobiologists in the UK is going to grow from these missions, even from all the data that we get back that needs to be analyzed. Okay, moving on. Greg has asked, what about the possibility of life in the clouds of Venus?
Ooh. That's an interesting question. I can't say it's one that I have looked into very much, but it's a very interesting idea. That's something, obviously the surface of Venus, we know, is under a lot of pressure and very, very high temperatures because of the thick atmosphere. But for something to be able to survive in the clouds, it would have to be very different from things that we know on the Earth. So it's not something that I actually know much about, but I do know that the podcast that I actually suggested at the end of this, it is mentioned the possibility of life in Venus's atmosphere. So maybe have a listen to that podcast and they will be able to answer that better than I can, sorry.
Well, thank you for that plug for our podcast. At the RAS Supermassive Podcast, which is going to be talking about searching for alien life with Professor Jane Greaves. So good question, Greg. Thank you. Here's one that we are familiar with, Rosie. Stephen Beard asked, do you think any life could survive on the moon?
So technically there is life on the moon. There's I'm sure a lot of you heard about tardigrades with a spacecraft that crashed into the surface of the moon, and they were left on the surface, but we don't think that they survived that. So tardigrades, basically, when they're stressed by as they would be in space, they kind of dry out and they go to sleep. They go into a tun state, we call that. And to be able to wake them up from that, they need some kind of water or rehydration, and they just wouldn't have that on the moon. And other than that, because the moon does not have an atmosphere to protect it, kind of similar like the surface of Mars, it's going to have a lot of this ultraviolet radiation and very, very low temperatures. So as far as we're aware, nothing could survive on the surface of the moon, but that's as far as we know at the moment. So there's always new work going to these things, and we're finding more and more of these extreme microbes.
Yeah, I mean, it's hard to say. I mean, we haven't spent a lot of time on the moon. It's the only other object in our solar system that humans have been on, but there's always more things to find out, more science to be had, and more exploration to do. Okay, so this is one of a good question that I'm really enjoying from Jim Mehta. He says, "Hi, Rosie. Have we found any organisms on Earth that can survive in a simulated Mars environment where all the conditions are replicated if they were on Mars?" And he says, "Great talk, and thank you."
We've definitely found organisms that can survive the high amounts of radiation and the desiccation on the surface of Mars. And I believe also the low temperatures at the same time. And that's an organism called Deinococcus radiodurans. But I'm unsure as to whether it can also tolerate the salt concentrations as well as that. So it's a good chunk of the different stresses on the Martian surface, definitely. But being able to test that out with more microbes and understanding more about if they can survive all of them, how, is very exciting, and it's something I'm very interested in.
Are you meaning like perchlorates that we have previously? I think with the Phoenix mission, the Arizona State University had was supporting. They had uncovered some permafrost on the surface of Mars, or just under the soil.
Yeah, so there's perchlorate salts and just lots of like there's high concentrations of just table salts like we find here, but the concentrations can be so high that the organism would have to be able to tolerate that much salt, and sometimes it can just disrupt their cell membrane that keeps everything in and keeps them nice and healthy, and that's not very good for them. So we kind of need to know how the different salts affect how they grow too.
Yeah, that's interesting. Tardigrades are so fascinating. I know we were earlier talking about tardigrades on the moon, but I've read somewhere about how tardigrades have this ability to kind of switch on and off hydration and dehydration, and I wonder if that ability kind of protects them maybe from a very high salinity environment. Anyway, maybe I should look into that research. Okay, Nova Neil asks, how does a living organism live in a 100-degree C environment, and how do biomolecules or proteins survive so they don't get denatured?
Yep, that's a really interesting question. So different types of microorganisms have different ways of adapting to these extreme environments. And quite often it does relate to their cell membrane, so the kind of envelope around the cell that keeps everything in. So as far as surviving in high temperatures, I'm not 100% sure on what that exact mechanism is that organisms tend to use, because most of the stuff I've been looking at has been looking at lower temperature environments. But for example, in a low temperature, organisms can have different adaptations. Like they can actually have what are called antifreeze proteins, which kind of develop within the cell and stop the interior of the cell from freezing when they're in a low temperature environment. So the organisms that can survive in those high temperatures will have some specific mechanism, probably to do with their cell membrane, that will help with that. And that will protect, obviously, as well, the proteins and everything else.
So amazing. It sounds like these other organisms have like these superpowers that I wish I wish we could have some of that. Okay, Anonymous requests a question here. Assuming that searching for life as we know it, so I guess life on Earth, is limited by current knowledge, how is the possibility missing life as we don't know it catered for in missions? So I don't know if understanding that question in another way might be like, are those Mars rover just looking for rovers that we know, like on Earth? How do they know what to look for? How do they know what life looks like on this other planet?
Yeah, definitely. So it's definitely a big thing in astrobiology, is that when we're looking for life beyond Earth, it's very Earth-centric. We have to go by what we already know about. And our only example of life is the life here on Earth. So when we are searching on other planets, we are looking for carbon-based life, similar to here on Earth. But that's a very interesting question. You raise a good point that what if life isn't the same as here on Earth? And we just have missed it because it's not what we've been looking for. And it's something really interesting. And work is actually going into that, to seeing if you were to use a different thing for a backbone of a molecule instead of carbon, and looking for biosignatures relating to that. So there is work and simulations going into looking at that, but currently we're very much just focusing on what we already know because it's easier to look for when we're on another planet or moon.
Yeah, one of the reasons why I studied geology, I was really interested in astrogeology, looking at the geology of other planets. And I was, surprised to realize that, well, you have to learn about the geology of your planet, learn what you know first before you can then study something else. So, and that's just all we can do, because until humans start exploring other planets or celestial bodies, we're not going to really know for sure. So, but I love all this remote science and all this data. It has been amazing that we are getting back from these robotic missions. Here's a question. Does the Drake equation serve any useful function in the study of exobiology?
It's a good question. It's more of a Drake statement, I guess, kind of saying about the estimate of the number of active galaxies that might be able to communicate within our galaxy. Again, the Drake equation is quite complicated to just go into on a whim, but I'm again going to direct you to the podcast, which is another thing that's actually addressed, is the Drake equation, and go into a bit more detail about that. So yeah, instead of trying to go into you without the actual equation in front of me, I'm going to direct you again to the podcast.
Wow. There are just so many factors that you can put into the Drake equation or not put into the Drake equation, and you're going to get different answers all the time. It's not an easy question to answer, and I look forward to what they might say on Supermassive Podcast. I'm sure they will. I'm sure they'll probably give something very similar. All right. So, an attendee whose name is Orion B specifically, they wanted to say they enjoyed your talk, Rosie, and their question is, even if we found life on some other planet or moon, how will the space agencies identify them that it is different from life? It sounds like a very similar question. Is there anything you want to add to that?
Yeah, I guess it's the same. If it's similar to life on Earth, then we will be able to detect it easily. If it's not, we might detect that there's something strange, like maybe the combination of molecules put together is very different from what we're expecting, and therefore it might flag up that we need to look into that more. But really, yes, it will be just, if we find something, we'll be looking for terrestrial kinds of life.
I wonder if, when I worked with Dr. Chris McKay at NASA, this is one of his questions that he normally gets. And he always talks about chirality, and that, life that we know it is one type, and that if we find something that's its opposite, that would say it's a different type. I don't know if that's anything that you've come across in your research.
Yeah, it's not something I've come across in my research, but it's definitely something I've heard in background information talks. And yeah, it's again that just it being that different that you can kind of realize that something's not right, but maybe we should look into that and see what it is.
Okay, great. So Grace Richards asks, they say, thank you so much, Rosie. Really interesting talk. And it was represented in a very accessible way. And I agree with them completely. It was a fantastic talk. Robin asks, where do you think that is the most likely place to find life other than Earth? Because that's an easy answer. We asked our fellows this question, I think, last year, and that's what they said. They said Earth. Anyway, what do you say?
That is a tricky question. So in my opinion, it would probably be, for current life, one of the icy moons, either Europa or Enceladus, because of the subsurface liquid water oceans. But we just need to be able to get out there to study them more. So with the Europa missions that are upcoming, we'll be able to find out a lot more. So I would say Europa is probably currently, with all of the missions we've got going there, our best bet for proof. But we do have all these missions going to Mars that are looking at subsurface environments. So we just don't know what they're going to find. So it's one of those things that kind of, whichever one you think changes with what's discovered and what missions are going out. But currently, in my opinion, I think Europa.
Europa. Okay, great. So Abby is telling us that tonight we can look out for the comet Neowise, which is absolutely true. We're going to be Neowise will be in the northwest part of your night sky, and actually, I think during the later part of this month. I think right about now it's in the early dawn hours. But definitely something to look up and observe if you get a chance, because we're not going to be able to see this comet come back around. Its orbit is like 6,800 years. So a few centuries later, you'll see it again. But of course, we won't be around. But she asks, do you think comets could harbor life?
Ooh, that's interesting. And also thank you for the tip about looking out tonight. So again, comets aren't one that I've really looked into. There would be extreme low temperatures involved with that for sure. But yeah, in all honesty, it's not one that I really know even where to start thinking about the habitability of a comet.
Well, then kind of the next two questions are very similar. So I'm just going to say them out loud, and you feel free to extend on anything. Grace Richards asks us a very similar question. Do you think life will look like us? And what do you think about looking for life that isn't like us? So we kind of already talked about that, but is there anything you want to add to that?
Again, just kind of emphasizing that microbial life is probably the most best bet for what we would find, rather than intelligent life like ourselves. And yeah, again, that it would be looking at terrestrial and using life on Earth as a basis for that.
And then the second kind of familiar question is, since we talked about comets, we've previously talked about life on meteorites. Also, can we expect existence of life on asteroids, so larger bodies, but not moons and not planets out in the solar system?
Yeah, again, I guess the speeds they'd be traveling at, and also it depends where they're traveling to. For example, if something was traveling towards a planet which had an atmosphere, it would burn up within that atmosphere, which would give such high heats. And any kind of microorganism, quite a lot of them that use photosynthesis, it would just stop their ability to be able to do that, and they would be destroyed. So I think it's quite unlikely because of that. And also, if it is just traveling through space, it's got all of the harsh conditions of the vacuum of space to deal with as well. So yeah, anything without an atmosphere or some kind of protective envelope from radiation is quite a tricky one to assess.
Okay, great. I'm going to go to YouTube now. Someone, thank you so much, who's an attendee here on Zoom, alerted me to there are questions on YouTube. So yeah. This is a great one. Thomas McDougall asked, what are your thoughts on theories that Earth may have seeded the rest of the solar system with life through debris from meteor impacts?
It's definitely an interesting theory. It's not something that I really think would have happened from my understanding of life on Earth and how it goes. But it's a very interesting theory, and I'm not exactly sure how to answer that question. Sorry.
And that's perfectly okay. We can't, I know we can't always know answers to all the questions, but they're really great questions. What shows is that they're so curious and they're very interested. Yeah. So I love that. That's really great. He also asks, if we found life, how would we establish whether it was alien or of Earth origin? And I know we've had this question several times already, but I think what he means, because he puts in brackets here, is it from meteor ejecta, or maybe it was brought in via the space probe? So sometimes we try to clean the rovers before they go out, the robots. Yeah. But I don't think they have to be 100% clean. So how do we make a distinction between those two?
That's a very good point, because we try and sterilize spacecraft or anything when it's traveling to another planet. But obviously, if something was missed, then we'd have that challenge of trying to differentiate. If we did find something, say on Mars, was it something we brought there, or was it something that was already there? And I guess the best bet is from these kind of experiments that we're already doing, looking at the kind of organisms that can survive in these environments on Earth. We have a good idea of the kind of organisms that are on Earth that can survive these kind of conditions that are on Mars. So if it was something completely different to that, it would be more of an indication that it was something from Mars instead of something brought from the Earth. And if it was similar to these organisms, then maybe it was something brought from our planet. But to find the exact fine details of that would be very, very difficult. So yeah, that's a really interesting question.
Yeah, I know we look to science for all the answers, but still in our experimentation, there's always this little bit of amount of doubt. And I think the great thing about science is we repeat the experiment to see if we get the same result, or someone else repeats it for us and see if they get their same result. And I think that was something that happened when we found liquid water in the subsurface of Mars, was that NASA might have seen some of this evidence years ago before they announced it, but they waited around until ESA did a similar observation to the area that they were looking at, and also found liquid water in the subsurface. And so then they write a paper and they say, okay, we have this amount of confidence to say, yes, we have found liquid water, you know, 1.5 kilometers under the surface of Mars. But it takes years for a sort of a consensus to come together, because I think there can be that sort of doubt and of you know, trusting your data. And I think it's a good idea to have another scientist, you know, do your experiment and see if they get the same result. Okay, still with YouTube, Christina asks, hi Rosie, it was a great talk, thank you so much. Do you have an opinion on the habitability of the Neptunian moon Triton?
Ooh. So Triton's another one we think could have a liquid water ocean. So again, it's got the same chances of habitability as Europa and Enceladus with that. We obviously know less about it because it's further away, so we've had less chance to actually observe it. But yeah, it's a good moon. It's a good chance that something, like, we do need to send more missions out there just to analyze it more, because Triton is an interesting moon in itself, with its retrograde orbit. So yeah, it's definitely one to look into.
Yeah, I don't know. That's a good point, Rosie. I don't know how much space, how often we've sent a spacecraft that far out into the solar system.
No.
Yeah, so that's a really good point. So last question on YouTube. They asked, could the asteroid belt have temperatures just right?
Oh, that's an interesting question. I'm just trying to think of that. So I guess if everything was close enough together and it's kind of getting pulled by Jupiter in one way and the internal planets another way, then there could be this kind of heating for things on there. This is all me hypothesizing. I don't know any of this for definite. This is just me trying to think if any of the temperatures could be right. Again, it would be things without an atmosphere, so they would very much need heating from some other kind of mechanism than heat being kept in from an atmosphere. So potentially, but I can't give you a definite answer to that one, sorry.
Okay, great. Okay, so we have about 3 minutes and maybe about 4 questions. I think we can get through all of them. How do you feel, Rosie?
Yeah.
Okay, cool. So Mauro, back to our Zoom attendees. He says, hi, Rosie. He's bringing attention to a paper. A couple of months ago, I read a very interesting article, Mechanisms of Water Extraction from Gypsum Rock by Desert Colonizing Microorganisms. Are you familiar with that? I can send you the link if not. And they go on to they go on to say that they find it quite fascinating that bacteria can extract water from minerals and rocks, and something they had never thought about or heard before. Could you comment on this?
Yeah, definitely. I think I've read that paper. I'm in the process of writing my thesis and I have so many papers open at the moment, but I'm pretty sure that's one of the ones that I have read, but thank you for putting my attention to it. And yes, the fact that bacteria can do that is really, really interesting. And it's, in fact, a research project that's going on as part of our lab. One of my colleagues is looking at biomining, which is microbes extracting these minerals from rocks and if they can survive from that. And even doing that on the International Space Station to see the effects of microgravity on being able to do that. So it's really fascinating that microbes are able to do that. And yeah, hopefully we get more studies looking into it.
Yeah, very cool question. I want to read that paper now. Thank you for the link. Okay, Rosalind, someone named after the ExoMars rover, Rosalind Franklin, asks, they say, hi Rosie, I'm really enjoying the lecture, but I have a basic question. What's the difference between the study of astrobiology and exobiology?
So as far as I can tell, they are the same. I think exobiology was founded first in America, and it was coined exobiology, basically looking at like beyond Earth. And then it just kind of got coined astrobiology as time went on. So I believe they are the same field. They just have different names because exobiology was around first.
Yeah, I agree with you that names change because nobody says astrogeology anymore, which was what led me to study geology in the United States, in California. I was really wanting to know about the geology of Mars back then, and this is in the early 90s when we knew very little. But nobody uses that term anymore. If you go to an old JPL site, they'll call it astrogeology, but I think now it's called planetary science or something. So yes,
Yeah.
Rosie's absolutely right. These names change quite a lot. I think as we know more, we change things. Okay, I think we're down to the wire. Oh, nope, someone else took a question. We'll get to it. Do you think that there are alternatives to DNA as the core genetic material? And if so, what do you think that might be?
It would be super interesting if there was. I honestly have no idea what it would be if it was something different to DNA. I'm pretty sure a few years ago, NASA released an article saying that they managed to model a different basic structure to DNA. But this isn't something that's been actually recreated in the lab. This was just done on a computer as hypothetically, could this work? So people are looking into what, if anything, could be used, and if so, what. But yeah, currently we don't know what alternatives to DNA there could be.
Okay. I think that's it. We're just 1 minute past 7, and just so that you know, the person who asked that question also said much kudos for being so honest and saying when you don't know things. And oops. My camera's on, and I agree. I think people who are specialists and anyone who really studies for a very long time, no one can really know everything. And I agree. I'm very respectful and very wonderful when you think we put scientists and people with PhDs up on a high pedestal, as they do deserve. But yeah, they're human beings too, and they can't know everything. But thank you so much, Rosie, for being with us and answering as many questions as you can. And I know that your research is focused on something really specific, and there's so much information out there, and we're constantly learning more. It's actually, I would say, very difficult to know to know everything, even if we tried. But we hope that this has inspired you to maybe look into astrobiology or other STEM subjects. And again, thank you so much, Rosie Kane from the University of Edinburgh, to be with us today. And if you want to say any last few words, Rosie.
And I just thank you for the great questions, and thank you everyone for tuning in, and make sure to watch the next bicentenary lecture, which is in a couple of weeks, I think.
Yeah, we're going to have one every month. We're going to have one every month, and I think one is coming up pretty quickly. But what we do have next Friday, thank you for this, other than our supermassive podcast, we're going to be live streaming from Jupiter, looking at Ganymede, transiting the face of Jupiter, following its shadow, and looking at the ionosphere of Jupiter. We're going to be doing some remote science with you as attendees, if you want to join us, observing from Mauna Kea. So we're going to be live streaming from 11 a.m. to 4 p.m. with scientists from the University of Leicester, also having Professor Emma Bunce, who's going to talk about Jupiter and the future JUICE missions, which Rosie Kane mentioned, and also maybe some of the data that came back from the Juno mission, as well as our RAS librarian, Dr. Sean Prosser, who's going to share with us some artifacts, some early observations of Jupiter that are always amazing to see, what scientists did and how they observed the planets with very young versions of tools that are just have now grown up to be so amazing today, have evolved to something like the Webb, fingers crossed when it gets launched, and like Mauna Kea or the ELT. So what is the future of astronomy? More things to come, more bicentenary lectures all the way up until December. And by the way, we are going to have one on the ELT, and we hope that you can join us again. For now, just want to say thank you so much, goodbye, and have a great weekend.